All right, this is basically a formal presentation of this recent paper by myself, Eric Trem from Deakin University in Australia, and Ben Haller in Cornell. I did the coral part, Eric Trem did the biophysical connectivity part, figuring out where the larvae would migrate. And Ben Haller supported the whole coding aspect of the model because he's the developer of the major software that we used for building up the model. So realizing that different people, different expertises could be watching this, let's make sure we are on the same page about corals. Corals are not rocks, right? So if you thought it would be a geology talk, feel free to leave. Corals are colonial organisms of the phylum Cnidaria. You can think about them as a bunch of tiny little sea anemones. Some of them actually do look like this, sitting together as a sheet and forming a clonal sheet out of themselves. And below themselves, they secrete this calcium carbonate skeleton, which actually makes the rock, which you know as a coral. And this calcium carbonate skeleton can grow sometimes in bizarre shapes, creating all sorts of branched and intricate forms that corals have on the reef. But if you look really closely, it's nothing but the whole bunch of little sea anemones sitting side by side together. One important aspect of their biology is the presence of symbiotic algae inside their gut cells. They're basically like chloroplasts, which still have a free living stage in their life cycle. And their function is exactly like a chloroplast function. They do photosynthesize and supply coral with energy. So this symbiosis is essential for their well-being, and many of you have heard of coral bleaching, which is disruption of this symbiotic relationship. Under stress, corals and algae forget how to talk nicely to each other, and the symbiosis dissolves. The algae leave the coral, and what you see, instead of the typical brownish color, which is given to the coral, by the way, by presence of algae, algae are brown, right? Because those are not green algae, they're dinoflagellates. So when they leave, you only see the calcium carbonate skeleton through the transparent tissue of the coral, and it looks white. So that's why it's called bleaching. This is a symptom of a very high stress. It doesn't necessarily spell doom and death. Corals often recover from this. However, if the stress does not abate, you can have die-offs on scales of the whole reefs, and this is increasingly frequent and becoming increasingly important factor in global coral decline, which we're having right now. So if you look at where corals live, this is the map of global coral diversity, and you see this big purple blob in the area which is called the coral triangle. It's kind of triangle if you're creative in your thinking. In the Indonesia central Indo-West-Pacific, this is the area which we model in our model. That's where the most corals are. This is how the temperature have already risen. Actually, already quite a long time ago, it was already quite noticeable increase. And you see that a lot of red blobs here corresponding to increasing temperature. And prior to just a few years ago, a lot of papers have been published saying that, look, if we just consider present day thermal sensitivities of corals, at which temperature they would bleach right now, and we project things into a future, we'll find that right in the middle where the coral diversity is the most, we'll have basically this big burnt out hole of uninhabitable environment for them, because it will be so hot, nobody will be able to tolerate and we basically lose this whole major biodiversity of coral reefs. So one important thing that these models assumed is that whatever corals are able to tolerate right now, these are the tolerances they will carry on in the future, and nothing will change. There will be no evolution whatsoever. And so under this assumption, the predictions are pretty dire indeed. But were they right at all assuming this? You often hear the argument that, well, evolution is such a very slow process. It takes thousands and thousands of years to evolve something. So probably it's absolutely not going to keep pace with the current rate of warming. This is the typical argument being made. I'll try to convince you this is not necessarily true, and most likely not true for corals specifically. So if there is one thing I would like you to take home from this talk, like one thing, this is it. Evolution is not slow. I try to dispel the common misconception, which is not just general public, but even professional biologists think of evolution as a slow process. There are multiple examples of evolution being able to produce completely different morphologies and physiological adaptations in as little as 50 years, from this marine stickleback fish to the freshwater stickleback fish. And even some examples are even more incredible, such that these green anoles populations in Texas evolve higher cold tolerance just as a result of a single bad winter, which was having a lot of winter storms. And so the population, which was just a year before, had a not such a not such a good cold tolerance, that they were all dying at about 10 degrees. But there was variation there, and after this intense selection, you get a population which is actually much more cold tolerant, just as a result of a single round of natural selection right there. And this cold tolerance persists throughout the year. So basically, this was one round of selection creating a dramatic adaptation to thermal environment, which is actually relevant to our subject today. How is this magic possible? So let me explain a little bit, just slide like picture by picture. Typically, people think of evolution as this. So you start with genetically homogeneous population, and in which a new beneficial mutation arises. And well, once it arises, it's beneficial. Natural selection allows it to spread through the population. And then nothing happens for a while until the next new beneficial mutation arises. And then this one spreads through population, and so on and so forth, mutation by mutation, which would be indeed very slow, because you have to wait for the new beneficial mutations to arise every time. This would be indeed bad and slow. Fortunately, this is not how things evolve in nature. This is how clonal bacteria in flasks evolve in the lab. In nature, there is this wonderful, especially in eukaryotes, there is this wonderful thing called sex, which allows creatures to recombine the mutations that they already have in their genomes and create new combinations of mutations together, novel, better adapted genotypes, without waiting for the new mutations to arise, just based on combinations of pre-existing ones. So this is a faster process because recombination happens throughout the genome every time somebody reproduces. So that's pretty cool. Still, this particular cartoon is not quite like how biology really works, because we all still start with the genetically homogeneous population. In nature, all populations are somewhere well along this process. So they already contain a whole bunch of different variants in them. So the proper picture would be like this. You start with already pre-existing mutational variation, which we will call standing genetic variation. This is the term which is very important for talking about evolution. Standing genetic variation is basically all sorts of variation which is genetically determined in population and can be inherited. So we also have recombination plus new mutations, but here the rate of evolution basically is determined just by extent of standing genetic variation and the rate of recombination. New mutations may not even be necessary. And so evolution based on this processes, that's the examples of very rapid evolutionary transitions I gave a couple of slides back about sticklebacks and anoles. That's how it works, really rapidly. You don't have to wait for anything to appear de novo. You just take what you have already, reshuffle it a little bit, and rapidly end up with a new adapted genotypes which can be picked up by natural selection. Okay, standing genetic variation is a very common thing everywhere. It exists for pretty much every trait in every organism, from humans to clams to corals, including standing genetic variation for bleaching tolerance, like you see here or here. In every bleaching event, even the most devastating ones, there were always some survivors, some corals which for that reason or another don't die. And these are the survivors which will give rise to the next generation. So you can see how if this is genetically determined, really, then you can have pretty rapid evolution. Well, so the question is, oh yeah, one more thing about rapid evolution. Corals actually have additional facilitating aspect of their biology. They exist not as one single interbreeding population, as you know, but many different populations, which are inhabiting quite a lot of environmental gradient. And these populations evolve on their own, adapt to the local conditions, and exchange migrants. This is called the meta-population. And this is like the previous situation with standing genetic variation, except we now have independent factories producing these standing genetic variation. It's like a bunch of independent working groups looking for solutions to future problems and exchanging notes occasionally. You can see how this can be a very efficient engine to generate potential adaptive genetic variation. So it creates a large common pool of adaptive genetic variation. When conditions start to shift, for example, everything warms up in this thermal gradient. Well, all you need to do is to reshuffle these already existing adaptive variants among populations and rescue the ones which are used to be cold and now they're hotter from extinction by just borrowing solutions which were already developed somewhere else in the interconnected populations. So redistribution of pre-existing adaptive alleles might be a very important aspect of evolution of corals. And this is the thing which we actually wanted to test with our models. We basically wanted to see how much new mutations, migration rates, and standing genetic variation would help corals adapt. Model setup, just to justify that we're doing the right thing. Yes, we know that indeed corals inhabit broad environmental gradients. Every species of a coral has a very broad distribution, usually covering even more than just the Great Barrier Reef, but more. And so even just looking at thermal variation, you already see a lot of variation here. They not only exist across the gradients, they do adapt to their local conditions. We know that for temperature, we know this from a simple fact that the way people predict the risk of bleaching is by the increase of temperature over the local value, not over some average species-specific value, but over the local value. That creates problems. So corals which bleach at 32 degrees here, the same species would bleach at 28 degrees here, as long as this number is by 2 degrees higher than its like typical temperature in the summer. So even from that, we know that local adaptation works. The question is, does it have a genetic basis, and can it actually be a fuel for natural selection? And us and some other people, well, this is the paper from 2005, which was spearheaded by Groves Dixon, my then graduate student and now postdoc, where we shown that if you actually take parental coral colonies from two locations, one hot and one cool, and you make babies using these parents, you have like babies which have only cold population parents, hot population parents, and the intermediate ones, and then measure heat tolerance among these larval families, you will find that the more parents you have from the warmer place, the more heat tolerant you are. And this is a clear, direct demonstration of importance of genetics in determining coral hot heat tolerances across these environmental gradients. Another important thing is that our model, as you will see, relies on a pattern of currents. And you might wish to ask yourself whether currents is a really good proxy for how coral larvae disperse. Maybe they swim, or maybe they are choosing where to end up because they like feel that they are in the correct environment or something like this. The good thing is that we have decent evidence that currents is the predominant force which generates genetic connectivity between the coral, in coral species. In this study, my graduate student, then graduate student, now professor at Boston University, Sarah Davis, she showed that for two species of acroporid corals in Micronesia, the basically distance which you calculate based on how currents go is a very good predictor for both of these species of how genetically divergent these populations would be. And last year, we managed to use next generation sequencing for 5 populations on the Great Barrier Reef to demonstrate that with some genomic magic, which I will skip the details right now, you can reconstruct the predicted migration rates between locations. And you can express them with these little arcs. You need to read them clockwise, and the thickness of the arc means the intensity of migration. And so this is what we see from the genetics, and this is what the currents are predicting. So it's not actually the same picture pasted twice, I kid you not. It's really very gratifying demonstration that whatever we measure with our genetics is actually makes some sense. And what we predict is current patterns. The take-home message so far is that if you want to model coral dispersal, all you need to know is the currents. Okay. So the model, I will basically skip this slide. This is for nerds who want to know how things work. I just mentioned that we use the software package called SLIM, and this is the Mondrian icon for this, which is individual-based simulation. We simulate each coral individual separately. It has overlapping generations, age-based mortality, and all good things. It has like the coral becomes adult at 3 years, and they evolve to local conditions using a pretty sophisticated quantitative trait loss, a genetic model. So glossing over this, please do ask me if you're interested in the end. I would love to like talk your head off with this. But basically, this is what it boils down to. Here is the map. Everybody loves maps, so I try to stick as many maps in my talk as possible. So this is what we model. We have coral populations across this central Indo-West-Pacific area where the coral triangle is. Populations are different size depending on the reef area in that particular location. They are set at particular local temperature, which is what the temperature is right now. You can see this as colors. And there are some migration rates between them, which are based on currents and some basic assumptions about larval biology. How long the larvae hold on in the plankton, how fast they develop ability to settle, and think what kind of mortality is, and things like that. Basically, you can predict the probability of a larva from a place A ending up in a place B. And that's basically the data which we put in the model. And yeah, and we let the model run for a very long time to equilibrate genetically, and we end up with the situation where you will have this map with mostly yellow reefs. This means it's good. Coral cover is about 0.6, which is kind of normal. When you see colors going into orange, purple, and then black, that means that the coral cover is dropping. It's really bad. So keep an eye out for these kind of events. So before warming, we basically end up with a situation like this, where on most reefs, corals are adapted nicely to their local conditions. Migration rates are going where they're going, and everything's nice and happy. And then we switch to global warming on at 2 different rates. We have a business as usual scenario, RCP 8.5, a really bad one, which predicts like more than 3 degrees C, I think, or more than 5. To be honest, I forgot how many. But this is a really, really bad warming scenario. And the RCP 4.5, which assumes pretty aggressive reduction of CO2 emissions in the world, and see if it makes any difference. So here is my movie of how the model runs. We start pre-warming, they're all the same. This model is RCP 4.5 with emission reduction. This is business as usual scenario, the bad one. And this number will indicate number of years since the start of warming. Minus 20 means, well, 20 years before warming. So let's see how it looks. There is also like El Nino type fluctuations in the model, so you will see some pulses of color. So in about 30, 50 years, you already see considerable declines in the business as usual thing. You may or may not be seeing how it pulses. That depends on your Zoom connection. But what what is pretty clear is that the rate of warming is really, really important in this case, right? Under business as usual scenario, most of the reefs in the area die, but not all. This is very surprising, and this is a very robust result which holds for whatever combination of parameters we tested. Some reefs emerge as very resilient in terms of their adaptive capacity to warming, including 2/3 of the Great Barrier Reef, Nova Caledonia, Vietnam, and some places in Japan. What is going on there? Hold this thought. First, let's see. Remember, the question was, why is that? Is that evolution based on standing genetic variation, new mutations, or migration, and how things are trading off each other? I mean, what do they contribute? So here is a 4 scenarios. In this one is the same as a business as usual I just showed you. This one is where we switch off any migration after the onset of warming. So there is migration before, so it starts from the same place, but then we say no migration as the warming starts, switch it off. Here we switched off both, migration and selection. So there is really no surviving. It doesn't matter whether you survive bleaching or not. Really, the next generation is not based on who survived the previous one. So in this one, we just switched off selection. Basically, think of this: no evolution, no migration, no evolution, and no migration scenarios. Let's see. Basically, what you will see is that the no evolution scenarios burn out really quickly and uniformly across the board. Interestingly, the no migration scenario does pretty well and pretty much undistinguishably from the base model until about the year 100. And then it gets into trouble. So from that we can conclude, let's actually watch this again. See how these guys disappeared completely? This because that every single coral in the simulation has died. So let's watch this again just to illustrate this point about the importance of migration. At first, the top 2 panels are pretty indistinguishable. And then after year 100, you will see that whoever survives here and there are going to go down as well. And they will be maintained in the base model. So our conclusions here is that evolution really, really matters. That's the most powerful thing. Evolution, probably based on standing genetic variation existing within the populations already. That really matters over the first 100 years. To make the evolution go further than that, you need migration to bring additional adaptive genetic variants into populations. So that's our conclusion here. And the thing which I'm not showing to you, and it's pretty surprising actually, but the model where we switched off new mutations during warming is indistinguishable from the base model. It doesn't make any difference whatsoever, which basically means that the whole process of new mutations arising in populations is completely irrelevant for what we're seeing. It's all based on pre-existing standing genetic variation in the metapopulation. Okay? So why things are surviving in some places? And we came up with 2 simple predictors which explain why some places are surviving better than the others. And predictors are, they make a lot of sense and they're pretty good predictors actually. One is how much immigrants the population gets from warmer locations, which basically quantifies its potential to absorb adaptive alleles from other populations. This is this graph. On the x-axis is the immigration rate from warmer places and the survival rate of reefs at 100 years and 200 years here. Pretty strong dependence on that factor. And likewise, present day temperature is also a very important predictor. Basically, the hotter it is right now, the worse it is. If you're already in a hot place, really you cannot evolve too much and you cannot draw on somebody else's solutions because you're already there at the boundary of where everybody else is. So by just using these 2 predictors, you basically can recreate the results of our model with good precision. So you don't need to actually run the complicated SLIM models, right? So this is the result to end all modeling. Well, anyway, it's another gratifying fact is that our predictors actually seem to correlate, well, not terribly well, but reasonably well and statistically significantly with actual coral cover changes observed by the year 2007 is data from this meta-analysis paper. So we actually are predicting something real here. Okay, so this is very cool. And we can conclude that, yeah, evolution based on standing genetic variation and adaptive allele exchange, but not on new mutations, is a very powerful force, which is going to be very important for corals. However, it doesn't mean that whatever we say is resilient or survives. It doesn't mean that they are safe. The problem is our model, just like any other proper model, is wrong, because it's deliberate abstraction from all other things except just the evolution. We don't model any ecology, we don't model storms, we don't model current changes due to or larval biology changes. We just leave everything out. The model is specifically designed to quantify the variation in adaptive potential and what affects it. So all the other things, basically it provides you with a baseline to compare to your actual observed data to see what the other factors are actually doing, right? It's the best case scenario. So very important for me to emphasize that whatever we say that the 2/3 of the Great Barrier Reef seems to have good adaptive potential, even 200 years in the future, by no means it implies that it's completely safe. No, we just need to make sure that it doesn't die from other things, which actually are multitude. Okay, take-home messages now. First one, most important, evolution powered by migration and selection, but not by new mutations, will matter a lot for coral persistence. Secondly, while most reefs are really at high risk under business as usual scenario, some reefs are remarkably resilient in our model, suggesting that they will maintain their adaptive capacity for a very long time. And that's certainly good news. We now know that we can recreate these results by just looking at two environmental predictors, immigration from warmer locations and present day temperature. That's a nice result. But also it's important to note that the model only looks at evolutionary potential. It is not a model of reefs in reality. To be honest, anybody who says that they modeled reef persistence in reality is probably lying or trying to sell you something. Every model is wrong, it should be, right? But some models are useful. That's the statistical mantra, right? All models are wrong, but some are useful. So this is a useful one, but it's wrong, right? Don't say that Great Barrier Reef is safe now. So even the most evolutionary protected reefs are not safe. We just need to make sure that they don't die from other factors. They're safe from lack of evolutionary potential. That's good, but other factors are still there. Okay, and with that, I would be happy to take questions. Yeah.
Thank you so much, Mikhail, once again.
No worries.
I would like to make a first question.
Let me find, wait a second, let me find my unshare button. There we go.
Okay. Yeah, great. So how do you, because this time I actually had more time to put attention to your talk and not just be concerned with the waiting room or with the emails. Last time I didn't get to enjoy your talk as much. So it made me think when you said that evolution is fast and everybody thinks that as it being slow, I count myself on one of these persons who thought that for evolution to happen, you had to wait many, generations or whatever. So how do you reconcile that and your findings with efforts that are undergoing now strongly to assist the evolution of corals? And so how do you have anything in the discussion of your paper that either sort of puts your results in that context, or yeah?
Yeah, thanks for asking, Sonia. Indeed, the assisted evolution is a term which is widely misunderstood. It means completely different things for different people. For general public and for even many biologists, it means that we're actually breeding supercorals in the lab.
Yeah.
So this is not going to work, and we have a separate seminar and separate discussion, separate presentation on how you would handle genetics if you want to do proper coral restoration. You need to maintain adaptive genetic variation. Standing genetic variation is the key to what I'm showing here. And so the assisted evolution that we can possibly think of is to help mix and match those adaptive alleles across the seascape. Basically raise the proportion of those immigrants from locations which are now representing future conditions. And this, according to our model, it would actually make help them a lot. If you yeah, we can even come up with the numbers of how much is actually necessary and sufficient to see a reasonable effect. So we're trying to incorporate these results now to actual recommendations for reef managers.
Okay.